Hi everyone, welcome to biology. I'm Miss Urban and this is our first lecture about what is science. What does it mean to know something? How does science differ from other ways of knowing? Science is tested against observation, it aims to disprove, and it doesn't care what the answer is to a question. Occam's razor states that when more than one explanation agrees with observation, the simpler explanation is usually right. And Albert Einstein stated, make everything as simple as possible, but not simpler. An observation includes new observations that you make using your senses or previous data that you learn through research. A hypothesis is a testable statement, often written in an if-then format. Experiments are processes used to test the hypothesis or further observations. A conclusion is your analysis of the results that are either support or reject your hypothesis. And a scientific theory are many experiments and observations together that develop a well-formed explanation of a phenomenon. An observation is the process of gathering information about events or processes in a careful, orderly way. Scientists collect data and use data to form inferences. The data they collect can either be qualitative, meaning things that are measured by quality. So color, texture, sight, sound, taste, smell, behavior, those are all qualitative observations. Quantitative are things measured in number. It's the quantity. So anything you are measuring numerically, length, width, height, weight, all of those things are going to be quantitative. And an inference is a logical interpretation based on prior knowledge or experience. Induction moves from specific to general. For example, these 6 elephants are sick. Therefore, I think all elephants in this population are sick. So you used inductive reasoning, moving from a small portion of the population and then making an assumption about the larger portion of the population. Inductive reasoning is used to generate a hypothesis, but it does not provide true knowledge. You use inductive reasoning to form your hypothesis, and then you're going to use deductive reasoning to test your hypothesis in order to show whether or not your hypothesis is supported or rejected. Deductive reasoning, or deduction, moves from general to specific. I think all elephants in the population are sick, therefore I'm going to test as many as I can, and I find that they're not all sick, so I can conclude my hypothesis was wrong. And deductive reasoning is the only logically valid type. Therefore, science can only disprove. Your hypothesis is a prediction based on inferences from observation. It must be testable to be considered scientific, and you make your hypothesis after you have made observations and done a lot of research on the topic. Your experiment is designed specifically to test your hypothesis, and they are going to attempt to disprove the hypothesis. The video that is linked here in the PowerPoint is actually a video from Monty Python about a group of villagers that are trying to determine whether or not a woman is in fact a witch. And they go through the scientific method while they're trying to figure out if she's a witch or not. And it's actually pretty amusing. So if you go to the PowerPoint slide, please take some time and click on the link. When you're writing your experiment, you need to keep in mind what the different variables in the experiment are, your controls, and your constants. The independent variable is the thing you're interested in and manipulate. It is what you change. The dependent variable depends on the independent variable, and it's what you're going to measure to determine if the independent variable had an impact. A control is the part of the experiment that's run with no treatment to determine if the changes would have occurred anyway, or if if there was a significant impact based on you changing the independent variable. And the constants are the things that don't change in experiment. So for example, if you were to measure plant growth in different colors of light, your independent variable would be the different colors of light. That's what you're going to change. You can test them under blue light, red light, green light. Dependent variable would be the plant growth. That's what you're going to measure. Your control would be having a group of plants that is grown under normal conditions, meaning the normal type of light. Your constants are going to be everything that you don't change, that you keep the same for every single group. The type of soil, the type of plant, how much water they get, how much light they're exposed to, the temperature they're grown in, the amount of fertilizer they get, etc. You want to keep all those things the same so that you know that you're just, the only thing that's different for all these is the color of light they're being exposed to, so you know that your results are valid. You're changing more than one thing for every experiment. Results, you're not going to be able to come to a logical conclusion from the results. So you just have to be very careful to keep your constants constant, and so you know what you're changing. When you write out your hypothesis, you're going to state them in an if-then format. If I do this, then this will happen. So you're going to talk about the independent variable first, what you're going to change, and how it's going to impact the dependent variable, or what you're going to measure. So your example is, if I change the temperature, then the plants will grow, with your temperature being the independent variable and plant growth being the dependent variable. I would argue that this example is not specific enough. How you need to talk about how you're going to change the temperature and how is it going to impact the plant growth. Aristotle was a scientist a long time ago, 384 BC to 322 BC. And he observed rats emerging from hay and rags in the corner of barns and concluded that the rats, which were living, arose from the hay and rags, which are non-living. He thought that flies could be produced by first killing a bull, then burying it with its horns protruding from the ground. Flies emerged from one of the horns, sawed off several days after burial. And his interpretation was that life may arise from non-living, and that was known as the theory of spontaneous generation, that living came from non-living. We now know that that's not true, and there have been many experiments done to disprove the theory of spontaneous generation. Living comes from living. One of the experiments that was done to disprove this was Reddy's experiment over here on the left. He had many mason jars, or many jars, and he put meat into the bottom of all the jars. He left some of them completely open, exposed to the elements. He covered some of them with a mesh type of cloth that had some opening in it so air could get through, but not big organisms. And then he sealed some completely tight. And what he found after waiting for a while is eventually the open jars had maggots on the meat. And maggots are fly babies, little fly larvae. The ones that were covered, both with the mesh and with the stopper, just completely sealed off, neither of them ended up with any maggots or any flies. And so his conclusion from that was that flies come from other flies. If flies are on the rotting meat, it is because another fly landed on the rotting meat and was able to lay their eggs. If you keep the flies off the meat, then there won't be any new ones. In the bottom right corner is a very famous experiment by a scientist named Louis Pasteur in the 1800s. He was working in the wine industry, and there was a lot of talk in that time amongst the scientists about there being some microscopic organisms that you didn't know existed and you couldn't see. And so they wanted to figure out what was spoiling the wine. Why was it going bad? Is there something that you could do to stop it? So he thought, well, if I boil it and kill everything that's in there, okay, kill any possible invisible creatures, then keep it sealed off. Will it not go bad? So created this special S-shaped flask, and it had this huge little tube for the opening where air could get in, but organisms could not. They would get stuck in that first curve of the tube, and they would not be able to get up and into the actual broth that he was testing. So when he boiled it, he left it be, and there was no growth. After he had killed everything and then he let it go and it wasn't exposed to anything, it was just sterilized, there was no growth, and so it preserved it. If you were to break the stem and expose it to air and allow different bacteria and microbes to get in there, then it went bad very quickly. It doesn't matter if it had been boiled before or not. He created the process of pasteurization, which was revolutionary for the food industry, and we still use it today. If you go to the grocery store, even if you go to your fridge, I am sure that you're going to find items in there that have been pasteurized, especially if you have any milk. Go ahead and look at the milk jug. It will say that it's been pasteurized. Here's a cartoon of Louis Pasteur looking kind of like a mad scientist and coming up with the process of pasteurization. A conclusion either rejects a hypothesis or it fails to reject a hypothesis. They never prove anything positively. Scientists publish their results from their experiments because it's very important to share information amongst the community. And other scientists will read that through the publication and repeat the experiment or refine the experiment and, hold each other accountable. It is never just one scientist always taking the word of somebody else. They're always double, triple, quadruple checking all of the work that is submitted. It goes through something called a peer review process where other scientists in your same field will pick apart what your experiment was and really try to reproduce your results or make them better or do the next step. And so in that way, science works as a self-correcting system. A scientific theory are hypotheses that have been repeatedly tested over and over and over again and have not been rejected, and they're treated with extra weight. So once a system of hypotheses providing a grand explanation of phenomenon reaches the point that it is almost assured it will never be rejected, we call it a theory. It is very difficult for something to become a scientific theory. And the word theory in science means much, much more than it does in just regular common language. Famous scientific theories include the theory of gravity, the theory of evolution, and the theory of relativity. So just a theory in science means that we are as sure of it as we are of gravity. Scientific laws are made up of facts. The scientific fact is an objective observation that can be measured again and again with the same result. Scientific laws are a statement of fact meant to describe in concise terms an action, often expressed as a mathematical equation. Typically, you will find your scientific laws in chemistry and physics, and most of them are going to be written with some sort of mathematical equation. Not all scientific laws are defined by equations, but most of them are. So a law is a generalization from a set of facts, typically written mathematically, and a theory is just an explanation of how and why things work. The law is what's happening, just real quick, here's your mathematical equation, and your theory is how it works, why it works, a whole lot more detail, a lot more in depth, a lot more evidence there. It's not just a simple statement. Have a wonderful day, and we'll see you next time.